Hello, everyone. I have the pleasure today of introducing Dr. James Doran, who's a postdoctoral researcher at the University of Bath. His research focuses on multi-scale mathematical models of infectious disease, and will be presenting today on his recent paper using CompuCell titled A Model of Tuberculosis Progression using CompuCell3D. James attended the workshop back in 2002 and the hackathon. And this particular work is a result of the project that he started during the hackathon that year. And I will pass it over to James.
Okay, good afternoon, everyone. My name is James Doran. I'm currently a postdoc at the University of Bath. And I will today be presenting some of the work I did during the course of my PhD, also at Bath. And specifically, I'll be presenting a within-host model of tuberculosis progression that we developed using CompuCell3D to compare to an original model developed by my former PhD supervisor, Bowness, who's currently my postdoc supervisor. So just to give a brief outline, I'll introduce some of the background on tuberculosis, sort of the severity of it, some of the interesting quirks behind it as an infectious disease, and how Ruth's group originally looked to model it. And then we'll go into some of the detail in terms of the interactions that their model contains, some of the biology that's been included within their model. I then won't go into any real detail on how CompuCell works, of course. The workshop will be telling you that itself, but more how we've used it to try and replicate that model as closely as we could. We'll then show the results that has been generated by our CompuCell code and compare them to results that we got using a sort of newer, more computationally efficient version of the original model presented by Ruth's group back in 2018. And then finally, sort of some discussion on the sort of differences between the original agent-based model and this CompuCell one, and potential areas for future work and future model development. So tuberculosis, commonly shortened to TB, is an airborne disease. It's caused by the airborne transmission of Mycobacterium tuberculosis, which for the rest of the talk I'll just be shortening to MTB. It's expelled by aerosols and can be as a result of any respiratory activity, so talking, breathing, coughing, etc. As the cartoon on the right shows, it's then aerolized and spread through the air. And if an individual who is susceptible then inhales these aerosols which contain the MTB within them, then they will potentially become infected by the MTB and experience TB infection. According to the World Health Organization, it is currently the leading global cause of death from an infectious agent. For 3 years, COVID-19 was the leading cause on this front, but since 2023, TB has regained first place in this regard. And in 2023 alone, it was estimated to have caused about 1.25 million deaths, and over 10 million people will be diagnosed with TB every year. It's believed that approximately 1 in every 4 people in the entire world will have been infected with MTB at some point within their lifetimes. Recent studies have suggested that this may be an overestimate in terms of how many viable infections there are that could potentially go on to develop into active disease. I believe that figure is closer to about 5 to 8%. But certainly it's estimated that at least 1 in 4 will have been exposed to MTB at some point within their lifetime. It's worth noting, though, that of those, about 90% will just have latent infection, so they won't develop symptoms, and they also won't be infectious and able to transmit the pathogen onwards. And it's only, fortunately, about 1 in every 10 infections that will go on to develop active TB disease and subsequently require treatment. The standard treatment is 4 different antibiotics that will be administered over the course of 6 months or 180 days. So one of each of the 4 antibiotics once a day, every day for 6 months. There have been attempts that have been made to try and shorten this treatment regimen, for example, to a 4-month length course of treatments, but these have not been successful. And some individuals may even require more than 6 months, potentially even up to 9 months of treatment, depending on the strain of MTB that they've been infected by. There is a vaccine, the BCG vaccine. Although this isn't perfect, it can help reduce infectiousness if you are sick and your susceptibility to catching the disease. Efforts have been made to make a better vaccine that potentially could infer some sort of lifelong immunity, but so far these have been unsuccessful. So as I said, about 9 in every 10 infections by MTB are latent, and the main reason for this is that in these cases, the MTB will be typically contained within a structure known as a granuloma. So the cartoon on the right shows what a granuloma, roughly speaking, looks like. It's essentially an aggregation of immune cells, bacteria, and necrotic, or in other words, dead tissue. So as you can see, what will typically be the structure of a granuloma where the disease has been successfully contained, you'll get some of these bacteria that are just extracellular, floating around within the lung tissue. There'll be these cells known as macrophages, which will have phagocytosis, effectively another term for ingested some of the bacteria, and so the bacteria are contained within these immune cells. Some other macrophages around the edges as well, and then on the outer ring you've got sort of the adaptive immune response of the lymphocytes like T cells that have arrived to the infection as well to try and help the macrophages that would have been part of the innate initial immune response. So normally in the case where containment occurs and the disease doesn't go on to the infection doesn't go on to develop into active disease, these structures will be fairly small, solid, well contained. The diameter will be typically less than about 2 millimeters across, and the bacteria will be well contained within them with no possibility of getting out. Obviously, this is a 2D representation of what in reality will, of course, be a 3D structure within the lungs. In the case of the remaining 10% of infections, it's worth noting, of course, actually, that there'll probably be more than one granuloma per infection. There's normally something on the order of about 10, but the biggest tends to be the most closely correlated to whether infection is latent or progresses to active disease being the outcome. So for the remaining 10% of individuals that will require treatment, this granuloma structure will tend to fail. The diameter will be quite a bit larger, potentially, than 2 millimeters, and it'll be a lot less well-formed structure with gaps that the bacteria can sort of escape from, disseminate throughout the rest of the lung tissue, and go on to spread further and cause eventually disease that will require antibiotics treatment. It's worth noting that some strains will also be resistant to the antibiotics that are used in the standard regimen, so they may require alternative antibiotics as well. So sort of more and more prominent issue is drug-resistant TB or extensively drug-resistant TB, although that's sort of beyond the scope of this talk, where I haven't really looked into antibiotics resistance for MTB, in this case, more just the immune response without antibiotics treatment. So latent TB, even if you are successful in containing the infection initially, months or even years after your initial exposure to MTB, you can go on to reactivate the infection and go on to develop disease. And this is normally observed in individuals where their immune response has weakened over time, so potentially after possibly co-infection with HIV, or from cancer treatment, or from just in general getting older and your immune system becoming worse over time, potentially. And even in the cases of successful treatment with antibiotics, it's still possible to then undergo relapse, potentially within the next few years after your successful treatment. And in addition, it's also possible that you could be reinfected with a totally different strain. And even if you're able to successfully be cured for one strain, another strain infects you and you potentially develop the disease all over again. So certainly no guarantee of any lifelong immunity. If you've initially been able to deal with an infection from MTB, you may still get it again later on in life. So the paper that was the inspiration for this work in my PhD was one written by my supervisor, Ruth Bowness, and a group of others. They developed an agent-based model of the formation of granulomas and the within-host dynamics of TB. And the main takeaway from their work was that where the initial cluster of bacteria is located within this sort of lung tissue where the granuloma will go on to form, relative in position terms to where the nearest blood vessels are and the general spatial arrangement of the surrounding blood vessels is the main factor, or one of the biggest factors in determining whether or not containment or dissemination will occur from that granuloma, and therefore how likely it is that an individual will have a latent infection or develop active disease. So in particular, those clusters that tend to be close to blood vessels, which in theory is where the immune cells are being recruited from, so therefore allowed for a short distance for newly recruited immune cells to get to the site of infection, were more likely to result in containment. Whereas if the cluster was a long way away from any nearby blood vessels and there's greater distances not only for immune cells to travel across, but also for antibiotics to diffuse across from the blood vessels to the site of infection, then these were more likely 2 leads to dissemination, a poorly formed granuloma structure, and therefore correlate more likely to an individual developing active disease. So the aim of our work was to try and replicate this model as closely as we could using CompuCell. Obviously, it's another Asian-based model, but it's slightly different in terms of its composition and sort of the cellular POTS framework that CompuCell effectively builds upon. So we wanted to see if, given these slight differences between the two frameworks, we're still able to get broadly similar results. To the best of our knowledge, it's the first TB model that's been developed using CompuCell3D. And in particular, whereas the original paper looked at a sort of a period of 12,000 hours or 500 days, so to capture the initial infection, potential treatment being required, reactivation, relapse, etc. Due to computational limitations, we've only been focusing on the first 200 hours post-infection. So the main focus of our paper was to see if we could get similar results to the original paper, just in terms of the initial innate immune response. So I'll just briefly discuss the sort of biological interactions that the original model included. Should note here, WHIDM is short for Within Host Infectious Disease Model. This is the name of certainly the most up-to-date version of the code that Ruth's group used in 2018. And it's worth noting that the biology of TB host pathogen dynamics is going to be a bit more complicated than what we present here. For instance, in the real disease, there's a range of cytokines that are known to be important to TB within host dynamics, but we've only considered one, a sort of generic chemokine that's used to capture chemotaxis. So in general, the immune cells will follow this chemokine and sort of move up the gradient from a low concentration towards higher concentrations to try and reach the site of infection. As you can see in the figure on the right, after the macrophages initially encounter the MTB, then they will release this chemotactic signal to attract other immune cells towards where the infection has taken place. So there's two main stages to the immune response. There's the initial innate immune response, and then there's a subsequent adaptive immune response. So in the innate phase, which is this sort of period on the left of the figure on the right, the macrophages will phagocytose the MTB and will secrete chemokines to signal to other immune cells to reach the site of infection. So over the years, because MTB has unfortunately been with us for a long period of time, the bacteria have learnt to resist the efforts of the macrophages to destroy them. So what will typically happen is that macrophages, after phagocytosing the bacteria, will just become infected themselves, and hence releasing these chemical signals as a warning sign that we really need help here with dealing with this infection. Over time, if a macrophage phagocytoses too many MTB, then it can burst and sort of release its contents back into the lung tissue. And then it leaves behind this sort of dead necrotic tissue. That's a sort of hallmark sign of a TB granuloma. Later on, after typically about 9 to 11 days after the initial infection, then the adaptive immune response will kick in. At this point, T cells will arrive at the site of infection. And within the original model, at least, their two main functions were to activate the macrophages. Activated macrophages are able to phagocytose a lot more bacteria without becoming infected by them, and therefore actually able to kill the bacteria off. And then T cells are also able to kill off infected or chronically infected macrophages, which also then kills the bacteria within them and leaves behind this sort of necrotic material with communication. So you can sort of see that flow through here. There's the initial encounter, phagocytosis, and releasing of chemotactic signals. This slow building up of a granuloma that, in theory, can then potentially split into another granuloma, and then we get the T cells arrive to the site of infection, and we get this sort of structure here with the bacteria contained within infected macrophages in the middle, more sort of uninfected macrophages surrounding that, and T cells surrounding that. So that would be an example of a sort of successful containment of the outbreak, and therefore the individual would probably have a latent infection if the granulomas were this sort of structure. In our case, as we only looked at the first 200 hours there, we haven't included the adaptive immune response. So really our main focus is on this sort of period up to this line here and this sort of nascent initial developing granuloma. So the two main outcomes that we can get will either be containment, and the innate immune response deals with infection already, or we get this onward dissemination, at which point the adaptive immune response will kick in, and if that still didn't bring the bacterial population down, then eventually an individual would probably be started on antibiotics treatment and diagnosed with TB disease. So within our model, we've defined containment to mean that fewer than 10 extracellular bacteria remaining at the end of our simulations, and dissemination being 10 or more are still left. So that's essentially all the biology that we're capturing. I'll now talk you through how we've developed that into a CompuCell3D model. So look at how the model's set up, each of the different cell types we've included, and all the different processes that our model captures. So we look at a 2 millimeter by 2 millimeter cross section of lung tissue. This, as I said before, like the sort of difference between a granuloma that's successfully contains an outbreak versus one that hasn't, is that the radius, the diameter will tend to be less than 2 millimeters for successful containment, and bigger than that if there's dissemination. So in theory, this domain size should capture the biggest possible granulomas that one is likely to see. We've initiated our model with a load of randomly placed blood vessels and resting macrophages with the exact numbers based on sort of biological literature. So any of these sort of blue cells are the blood vessels. I don't know how clear that is, but these are blue. And then any of the yellow ones are the resting macrophages, which are sort of just there in general within your lung tissue, even when there's no infection, just as a sort of background level, just so that they are there to respond if an infection does take hold. And as you can see, in the middle of the domain, in this case, we have our red and green cells. So these are the extracellular bacteria at the very start of the simulation. In this case, they were centrally placed, but in general, we start our simulations off with them being replaced randomly throughout the domain. It's been found typically that the infecting dose will be on the order of about 10 to 100 mTB. So we started all our simulations off with 6 fast-growing and 6 slow-growing bacteria. So I should point out at this point that we've used 2 phenotypes of MTB bacteria. This is based on the idea that it's been found that MTB, when it's found it's in an inhospitable environment, so for instance, there's a lack of oxygen, they'll enter into a sort of dormant state, they'll reduce their metabolism and their replication rate, and it seems to allow them a greater chance of survival when the environment is not favorable to them. And in particular, the slow-growing dormant state is the sort of bacteria that Ruth's original paper in 2018 suggests will be the ones that are left when we see these instances of disease reactivation or relapse after treatment. So it's worth keeping 2 different phenotypes in just to see if we see the differences in the sort of fast-growing state versus this dormant, slow-growing state at the end that the original paper suggested we should. So we've included all these different cell types within our model. Obviously, you'll be used to the idea of there being medium within CompuCell models now. And then beyond that, we have blood vessels. These are sort of statically frozen in place. And through them, oxygen is secreted into the domain, and any immune cells are recruited through the blood vessels. And then have these 2 phenotypes of MTB. Depending on how much local oxygen they sense, they'll either be quickly replicating or more slowly. We have 3 different types of macrophage here. So resting is the initial state that they're in when they have yet to phagocytose any bacteria. Once they've phagocytosed any, they enter this infected state. And once they've phagocytosed at least 10, they become chronically infected. The main difference between these 2 just being the rate of consumption of oxygen that they each perform. Then once a chronically infected macrophage has phagocytosed at least 20 bacteria, then they burst. They release the bacteria back into the domain, and they leave behind this caseum. Also, if either of these 2 just die naturally, they'll leave behind caseum, so this sort of dead tissue that we have frozen in place. And you'll also note this wall cell type as well. It's may sound slightly odd. Obviously, there aren't actual literal walls built into a biologically realistic granuloma, but this was to impose no flux boundary conditions on our model, as we found during the model development that some of the cells would stick to the boundary and not come free within our CompuCell framework. So by implementing a sort of wall type and just setting the contact energy such that they effectively push the cells back into the domain, we could enforce our no flux boundary conditions in trying to replicate the original model. So we have our 2 main chemical fields. One is oxygen, and you can sort of see a snapshot of how the oxygen fields may look over time on the right. And then we also have the chemokine for the chemotaxis. So both of these will evolve onto these reaction-diffusion partial differential equations. And it's also been noted that the oxygen tends to diffuse more slowly through granulomas. So the model set up such that the diffusion rate is reduced if the oxygen is within what would be considered the area of a granuloma. So any sort of infected macrophages, chronically infected macrophages, or caseum. The oxygen will get to the site from the blood vessels. So as you can see, if there's a lot of blood vessels together, you'll get these spikes, the sort of red bits in high oxygen concentration. And if there's no blood vessels anywhere near, you get much lower oxygen concentration, like the blue areas. And then this was then consumed by the macrophage and the MTB and is responsible for causing the MTBs to change phenotypes. And the chemokine will be released by any infected macrophages and decay over time. And the cells will move from low concentrations to high to get to the site of infection. Let's quickly go through some of the cellular processes. So to recruit resting macrophages, if we look at this sort of blood vessel shape, so it'd be a 5 by 5 within the CompuCell domain, volume of 25 voxels, and it checks each of its neighboring boundary, the boundaries lattice sites next to it. So this one, for instance, would check each of these 8, of which 5 are going to be on its boundary. This one will check each of these 3 on the boundary, and so on. So the numbers indicate how many times each boundary one is checked. If there's a free space, then in theory, with some probability, a new resting macrophage can be recruited into, say, this lattice site. It will start with a volume of 1, but it will be given a target volume of 25, and it will quickly grow to reach that target volume over the subsequent Monte Carlo steps. We also have our cell state transitions. So the bacteria will check their local oxygen concentration, and if it's above a certain threshold, they will, if they were slow growing, become fast growing. If it was below a certain threshold, then had they been fast growing, they become slow growing. And if the oxygen is just between these 2 thresholds, then there's no state, no change in the state. If they were fast growing before, they stay so. If they were slow growing before, they stay so. We also have the macrophages will change state depending on how many bacteria they've currently phagocytosed. So if it's none, they're resting. If it's above this initial, if it's below, sorry, this transition NICI, which sort of stands for the number to move from infected to chronically infected. If it's below that, it's infected. And if it's phagocytosed more than this, then it's chronically infected. And if it's reached the burst point, then it becomes caseum and it's this dead material. The cells then also die. So the macrophages will naturally die after a certain period of time. And if they're not resting, then if they are resting, they disappear. If they're chronically infected or just infected, they become caseum. And then, of course, they can also die through bursting if they phagocytose too many bacteria. And then the replication takes place as well. They grow until they reach double their initial volume, and that'll be after a set number of Monte Carlo steps, depending on if they're fast growing and slow growing, at which point they divide into 2 cells, and all the sort of attributes of the parent is copied over to the new child MTB that's been developed. That I think might be a good place to maybe show you perhaps how the code works in theory, because I appreciate I've spoken for a while. So if I quickly pause my talk and I'll perhaps show you a screenshot of what's going on. And I'll quickly share with you a simulation that I've been running from earlier. If I can. Perfect, here we go. So what you should be able to see right now is a simulation that I've just sort of ran to completion. So on the left, you can see the cell field of the model. In this case, the outcome was containment. And you might just be able to see in the top left corner sort of coordinates around 400 on the y-axis, 100 on the x. There's a collection of a number of infected and resting macrophages, which sort of indicates that we've wound up with containment, and this is where the granuloma is formed. You can also see in the chemokine field that there's this small buildup of chemokine that's been released. So as you can see, all the macrophages have sort of clustered around this high concentration of chemokine and reached the site of infection and been able to contain it. And then the oxygen is largely dependent on the concentration of blood vessels. So there's a lot in this sort of 100 x-axis, 100 y-axis area in the bottom left corner. And then other areas like the top left corner, there's been very few blood vessels, so there's almost no oxygen concentration up there. So this is how a simulation would probably look at the end of the runtime. If I shrink this back down and I restart, then you can see what it would look like at the start. These simulations tend to take about half an hour, so I won't do the whole one, or just leave it running for a few minutes and you can get an idea of how it evolves. I'll restart. So just a quick step. Okay. So as you can see at the very beginning here, all the bacteria are clustered on this sort of left-hand edge of the domain. So sort of y coordinate about 250, x coordinate about 50. Right next to a blood vessel, so there's a good chance that there will be a containment as the final outcome. You can see that blue blood vessel right next to the green fast-growing bacterium. You've got the yellow macrophages randomly dotted around, the blue blood vessels randomly dotted around. So that's the initial condition in terms of the cell state. There's also going to be the chemokine, obviously at the start, there is none of the cells are infected yet. This will stay 0 until the first phagocytosis takes place. And the oxygen at this point, very low concentrations, but you can see at the sites of the blood vessels, the very first secretions have just started to take place. So this is paused on the very first step. If I run it on, you'll see the model slowly start to evolve. It's fairly slow relative to some CompuCell models, so we won't necessarily see an awful lot over the next few minutes, but you can sort of start to see some of the things are happening. The oxygen concentrations in the blood vessels are now slowly starting to be secreted outwards, so these fields are starting to build up. Chemokine is still 0 because we have to have any phagocytosis. And in terms of the cell field, the bacteria sort of clump together at this point, but they'll slowly start to increase in volume. The green ones will grow a lot more quickly than the red ones as they're the fast growing phenotype. So in theory, faster growing MTB should replicate sort of every 15 to 32 hours or so on average, whereas the slow growing ones, it's more like 48 to 96. Each Monte Carlo step in our model, we calibrated it to get all the processes to act at the right speeds. And we found that the best time step size was about 24 seconds per Monte Carlo step. So it takes a fair few Monte Carlo steps for the certain number of hours to have elapsed. We set it up so that the bacteria won't change state until 300 Monte Carlo steps have gone in, just to allow the oxygen to have developed enough that we get a sort of fairly representative idea of the field. And as you can see, sort of near here, there's the one blood vessel secreting an amount of oxygen. So the fast growing one has stayed fast growing as there's enough oxygen nearby that it doesn't drop. All the rest are a bit too far away from any blood vessels, so they've changed to a slow growing state. And then over time, if the oxygen increases enough that it goes above the O high threshold, then some of these may go back to being green. That green one's probably unlikely to change now, given that it's near enough to a blood vessel that the oxygen level is never likely to drop too low for it. Hopefully we'll reach a point where a macrophage gets to the site of infection. Obviously at this point they're all just moving around randomly, so there is no guarantee, of course, that an infection event will take place, but hopefully just through pure randomness, eventually one will reach the site of infection or potentially be recruited from this blood vessel that's right next to the site of infection. So I should note that the bacteria themselves don't move, they're all frozen in place. This is sort of been found to be the biologically most realistic behavior for the bacteria, that they don't move about the lung tissue. They sort of just replicate once they've sort of lodged within a certain spot. The macrophages move through sort of two different means within this code. There's the chemotaxis once there's a chemokine to be sensed. But in the absence of that, then they move around due to the sort of external potential module, which allows them to just move with a certain amount of sort of force applied to push them in a certain direction to sort of that intrinsic motility that we would expect these immune cells to have. So apologies, it's not the most interesting simulation right now. We'll leave it running for a few minutes. If nothing's happened by 25:2, I'll just go back to my slide. But hopefully we'll see a phagocytosis event occur, and then can sort of show you that the chemokine does start to release and signal others near it.
So while we
Exactly.
wait, could you answer if this external potential, is it a biased force or just a random force that's applied on these macrophages?
Yeah, so it's based upon the direction that they're currently traveling in. We set the external potential up to be based on a sort of circular distribution. What was the name? Von Mises distribution. That was the one. So it's uh, the mean of the force. Oh, sorry, the mean of the direction of travel should be the same direction that you were traveling in initially. So the most likely outcome is you keep traveling with some sense of persistence, but then equally with some non-zero probability, the force may change to push you in a slightly different direction off from the direction you're going in. So that will partly depend on the dispersion parameter that you chose. If you have like a zero dispersion parameter, then you'd have an almost uniform distribution, and the force could be pushing you in any direction within the sort of 2 pi radians that you could be going in. Whereas if it's a very, very high dispersion parameter, then it's essentially a very, very, very steep normal, almost to the point of being almost like a direct delta distribution, where if the dispersion parameter is high enough, it's essentially almost guaranteed that the force will just be pushing you in the same direction that you're already traveling. So once this direction of travel has been chosen based on its probability distribution, the force is then going to be based on the cosines and sines of that in terms of the x and y components of the external potential. So for the most part, you should see macrophages largely traveling in the same direction, but every now and again having the possibility of slightly changing course. And then obviously the other forces like contact energies and volume constraints also impacts their movement as well. It might be worth coming back to that in a bit because nothing much is happening. So I'll keep going with some of my slides for now. But if anything interesting happens, then we can come back to that. Let's go back to my slides. So to go into some of the results, the first thing we had to do, given that this is a stochastic model, no one run is going to be representative of all the simulations that you could ever run with this. So one thing we had to do was run a consistency analysis and just try and figure out how many runs we would need on average until one batch of n runs, say, would be essentially no different to another batch of n runs. So the main thing we were checking is that if we ran say 300 simulations, would the total amount of bacteria within the domain on average be much different from another run of 300? And in the end, that this plot on the right is effectively showing that 300 leads to very small statistical differences in terms of the average number of bacteria left, whereas for the other distribution sizes that we tried that were smaller than that, so 1, 5, 50 and 100, these differences were deemed to be too large between any one set and another. So we ran 300 simulations for each of our results, as that seemed to give a fairly consistent averages and sort of standard deviations for our outcomes. So this is some of the maths behind it. I won't go into it in too much depth, but if you are interested, then this was taken from a book chapter by Hamish, Strasiev and Powerthill from 2021, and the references at the bottom of the slide. So as I said, 300 repeats, what this plot on the right is effectively showing us should mitigate any intrinsic uncertainty of the stochasticity of the model. And this was also found to be the right amount for the original agent-based model by Ruth's group, WHIDM as well. So all results were based on 300 simulations and just averaging from those 300 repeats. We then did a robustness analysis, so looked at some of the key CompuCell parameters. So the contact energy between bacterium and macrophage would influence how much phagocytosis was taking place. The chemotactic attraction coefficient was what we called effectively the lambda for chemotaxis. This von Mises distribution that I mentioned, the dispersion parameter determining how steep or flat the probability distribution was over the sort of whole circle of directions that the force could be pushing you in. And then elasticity coefficient was our name for lambda volume, sort of how much the cell wants to maintain its current shape versus being able to slightly deviate from that. So in terms of the contact energy between a bacterium and a macrophage, the default value that we used was 10, and we also explored all the multiples of 5 going from 0 to 20. Found that for most of these, there were very, very small differences in terms of the total bacteria left at the end, as you can see on the right. But for 20, there was a slight statistical significance in terms of the results, which is not the most visible from our summary plot on the right. But it did give us sort of thought of, okay, if we tried sort of orders of magnitude larger contact energies, how would that change the results? And as you can see, it does have quite an impact. So when we consider different multiples of 10, powers of 10, then using contact energies of 100 or 1,000 between the bacteria and the macrophages led to significantly higher bacteria numbers at the end of simulations. And in theory, this, of course, makes sense. If it takes more energy for a bacterium and a macrophage to make contact with each other, it will happen a lot less often. And in our model, phagocytosis would only occur when a bacterium and a macrophage made contact with each other. So setting this contact energy to be too high simply stopped phagocytosis from occurring effectively and allowed the bacteria to reach incredibly large population sizes at the end. So our model is clearly not completely robust to the choice of this contact energy if we set it above a certain threshold. The next one we investigated was the lambda chemotaxis parameter, and effectively from the default value of 2,000, checking all the other multiples of 1,000 from 0 to 4,000, there was no substantial difference in the results. In theory, we could have checked for higher values as well, although the literature suggests that having sort of these lambda values for any of these processes being too much larger than the sort of temperature default parameter tends to cause the cells to break up due to the forces being applied to them. So 4,000 was felt to be a realistic upper limit beyond which, yeah, potentially the whole behavior of the model starts to break down. So it appears that our model is fairly robust to the choice of the chemotactic attraction coefficient. Although, of course, you can sort of see that if it's 0, it's not quite statistically significant. Well, it's not statistically significantly more bacteria, but there's a little bit more bacteria than when we use the default values. So there's some small differences, but effectively negligible effectively. The dispersion parameter of the von Mises distribution we then investigated next. The actual default we used in this case was 99, so we approximated that with 100 and then looked at 1,000, but then also 10, 1, and 0.1. So the nearer to 0 you are, the more like a uniform distribution it is in terms of the external potential being applied, and the bigger it is, the more like a very steep normal distribution it is. So you can see, compared to the default value, if we go an order of magnitude in either direction, the bacteria results, sort of the ones on the right of the summary plot, are fairly similar. But if this is too close to a sort of uniform distribution in terms of the force, then effectively the macrophage could be being forced to completely change direction from where they're currently heading. This seems to effectively almost cause them to be frozen in place if there's too many changes in direction over time. And that appears to just make them a lot less effective to phagocytose the bacteria, and we see much bigger populations resulting as a result. And you can see the differences there. There's a very large statistical significance in differences between a low dispersion parameter for this distribution and the sort of higher ones that we use by default. And then finally, the elasticity coefficient. So the default value we used was 10. And you can see the model is the least robust to this, as any change in magnitude sort of by a power of 10 in either direction drastically changes the behavior of the model. So you can see that if lambda volume is set to 1 or 0.1, the cells have such little initiative to sort of maintain their volume that they just wind up fragmenting and then just dying off. So we get no bacteria left at the end of any of these simulations if lambda vol is too low. But this isn't because of a good immune response, because also all the macrophages have also completely disbanded and scattered to the four winds. So biologically speaking, it's not a very realistic outcome. Equally, if we set lambda vols to be too large, so 100 or 1,000, then we get the macrophages are so desperate to maintain their preferred volume that they basically just stop moving. And therefore, phagocytosis is much less likely to occur because they will never be moving to the site of infection, as all of the energy calculation in the Hamiltonian is going towards this cell wants to maintain its current shape. So in that case, we get these much bigger populations of bacteria left at the end, as phagocytosis happens much less frequently when the macrophages effectively stop moving. So in terms of the parameters from a CompuCell point of view, our model was fairly robust to the choice of lambda chemotaxis, but less so to the contact energies beyond a certain amount, or the dispersion parameters of the von Mises distribution, and so to an extent less robust to the external potential choices beyond the point, and were very much not robust to our choice of lambda volume. This clearly needed to be a certain value, and that was it. So having done this, we then actually tried to see, would we get the right proportions of containment versus dissemination in our simulations? And we were able to calibrate CompuCell fairly successfully in this regard. So as you can see, approximately 90%, give or take a small amount, did lead to containment and a proper granuloma forming, and 22, so again, just under 10%, led to dissemination, so granuloma not successfully forming to maintain the low bacterial populations, and in theory this would correlate to someone developing active TB. So these results correlate nicely with the proportions of latent infection versus active disease that we would see in reality. And in the sort of more computationally efficient version of Ruth's code, we got similar proportions, although I mean, I say 10% leads to active TB. In general, though, it suggests this is plus or minus 5% either way. So this was sort of more borderline, but in the end, we did get the right proportions in the more up-to-date version of her code as well. So both the codes largely agree in terms of the proportion of containment v dissemination. What we did find, though, which was interesting, was that the spatial organization was no longer impacting containment in either of these models. We think the main reason for that is the fact we were only able to investigate the first 200 hours. So our initial hope was to run the CompuCell model for the full 12,000 hour range that the original paper investigated, but we found that simulations were lasting too long to complete these simulations in reasonable time. So our main focus was on the first 200 hours. Obviously, at this point, there's no adaptive immune response. There's no T cells being recruited from the blood vessels, and there's also no antibiotics entering the domain as well. So our hypothesis, therefore, is that these are the main things that will therefore make spatial organization important in determining whether containment or dissemination occurs, rather than just the initial innate immune response and how easy it is for newly recruited macrophages to get to the site of infection. So the summary plots of our different bacterial populations are shown here. So in the CompuCell model on the left and in Ruth's up-to-date version of the code on the right, you can see in this case, this is the number of fast-growing bacteria left in the domains. There's sort of limited qualitative agreement. There's quite different orders of magnitude in the end. It proved quite difficult to calibrate the models in this sense. The results from Ruth's original paper, we were able to calibrate CompuCell3D quite effectively, but then for reasons that we're not entirely sure of, the more computation efficient version of our code more recently, it was harder to get to match the previous two sets of results we were trying to calibrate all three to effectively. So it's trying to hit a moving target in a sense, which was difficult. But we do sort of see this initial drop off in the fast bacterial numbers, and they stay lowish for most of the domain, although they are starting to rebound by the end of both to a much larger extent in the up-to-date version of Ruth's model. The slow bacteria numbers as well, for the most part, on average, stay low, although occasionally you can see from the dashed lines for the 95% confidence intervals, on occasion there will be some left at the end in the case of dissemination. And there is sort of the start of a rebound effect after a certain amount of time where macrophages start to die, release their contents, and the slow bacteria get released back into the domain again. Again, from the sort of quantitative point of view, the orders of magnitude are a little bit different between the two models. And then finally, the intracellular bacterial load, as you'd imagine, the average number of these slowly increases over time, the average being sort of just above the initial 12 that we initialized the domain with, to reflect the fact that some of those may have replicated, and of course some dissemination ones will have seen more phagocytosis occur because there's a lot more bacteria to deal with. So for both of these, that average rises over time, as you'd expect, but again, the sort of orders of magnitude are a little bit different. We were sort of surprised to see this, so we did just check if macrophage numbers were consistent between the domains as a possible explanation, but in the end, the average amount of macrophages left at the end is fairly close to the 105 that we initiate the model with. So it appears that whenever macrophages are naturally dying, we're recruiting them back with a close enough probability in both models. So the sort of the feeling was in the end that we were able to generate sort of qualitatively similar results. We sort of get similar structures to the granulomas that were left at the end. And from a quantitative point of view, we were able to get the right proportions of dissemination versus containment, but there were nonetheless orders of magnitudes of difference in terms of the amount of bacteria that were left. In part, we wondered whether this may be due to sort of the nature of CompuCell, as it's quite a sort of damped model where we sort of minimize the total energy. There's sort of more of a bias perhaps towards these sort of low energy stable configurations where not many cells will have had the chance to sort of replicate for there may not be much variation in the final outputs of a simulation that's sort of moving towards this sort of low energy state. Whereas obviously with TB infection, we could get quite volatile outcomes where there's no bacteria left, or where there's a huge amount left where we've got an outbreak, which Ruth's newer model sort of captures more compared to her original model and the CompuCell one that we have. It is interesting to note that other CompuCell models that have been compared to alternative agent-based implementations have also got similar outcomes. So in this case, this was a cancer oncology model where the original authors had an agent-based model and tried to replicate it with CompuCell. And similarly, they were able to sort of get qualitatively similar behavior, but the actual quantitative outputs were slightly different between the two models. So in general, it sort of highlights the difficulty that comes with trying to replicate one type of model approach with a slightly different structure elsewhere. In general, we found that the CompuCell framework was very easy to use. Obviously, the drop-down code snippets are very easy to implement. Certainly, it's quite an intuitive model to build up if you haven't sort of had much previous modeling experience, or if you're quite used to Python, say, then it's fairly easy to understand how CompuCell works and how models can be built with it. It would be interesting, though, to sort of explore some of the key parameters, as our sensitive analysis, of course, showed that if you don't set the initial sort of CompuCell specific values for the sort of lambdas of the, like lambda chemotaxis, well, lambda chemotaxis was okay, but lambda volume or the contact energies, if these aren't calibrated correctly, then you can get widely different behavior, sometimes not very biologically realistic behavior. So sort of more investigation into some of these key parameters would be, I think, beneficial. And the no flux boundary conditions may be one of the reasons why our results are is possibly a model artifact of our framework that's maybe not the most realistic, of course. It's preferable to periodic boundary conditions, and the idea of a wraparound granuloma doesn't make a lot of sense realistically. But equally, if the bacterial cluster starts too near to the boundary, then there's a risk that a granuloma can't properly form around it. And so future work could explore trying to place that cluster more centrally, with still having randomly located blood vessels and resting macrophages initially, to see if we can still get these right proportions of containment redissemination without the risk that a granuloma can't form, purely because of sort of the modeling choices that we made with the no flux conditions. So to summarize, this is the first within host TB model that's been developed using CompuCell. We found that our model was a little bit sensitive to certain key parameters like the lambda volume, the external potential or the probability distribution influencing the force, the direction of force from the external potential module, and the contact energies. Our model was quite sensitive to some of these parameter choices. However, in general, we think that there was a reasonable amount of cross-validation for WHIDM. We got qualitatively similar granulomas, and in terms of containment redissemination, we were able to get the two models to agree fairly closely. But in terms of the amount of bacteria left at the end, there was sort of a bit of a difference in terms of the orders of magnitude in that regard. And the interesting outcome from both models was that the spatial organization that was found to be important in Ruth's original 2018 paper was no longer important for just the first 200 hours, which sort of gives weight to the idea that that spatial organization is more important from a point of view of recruiting immune cells from the adaptive response for T cells and for antibiotics to diffuse in from the blood vessels, and that being easier to do if the blood vessels are nearer to the initial cluster. So I'll just quickly see if there's been any progress with our simulations, and then there's a I'll sort of talk you through how I've developed the code, and then in theory there's a sort of follow-on task for anyone that's interested. So you can see in this simulation, none of the macrophages have yet reached the site of infection. So there's been a little bit of replication in here. That is slightly more than the 12 bacteria that we started with. I don't know if you remember, but the screenshot I showed you earlier on in the talk had shown an instance of containment. So this shows that you can get different outcomes, and in some cases you will get dissemination, even when the cluster is near to this blood vessel. As we said, the spatial organization no longer appears to be as important for the first 200 hours. So I don't know how many. Yeah, I mean, we're only about a third of the way through, so I won't keep showing you that for now, but I will talk you through how I wrote the code. And then we can look into a toy model version that's sort of a bit simplified, and there's a few things that you can do if you're interested.
Do we want to pause for questions really quick on the presentation slides itself? If anyone has any questions on the presentation shown by James, please feel free to unmute and ask. If I set this up that you can't unmute, please just type in chat and let me know.
I also forgot to share brief acknowledgements to the workshop organisers. I had some hackathon teammates that helped with the initial development of this code. I'm an part of my PhD supervisor team, and then we have to run simulations on the supercomputer with the University of Bath Research Computing Group. And if you are interested in more details, also here's a QR code. You can scan that in, and you can access the preprint on archive. So I am happy to take questions.
I can ask the first question.
Sure.
So, of your distributions, you found sensitivity in mostly the von Mises and the elasticity coefficient, your lambda-val. So the lambda-val, not very surprising, it effectively can free cells. Did you look into just changing the volume size of the cells and how that would change? Because that's the other corresponding component, and the Hamiltonian contribution. And the other question is, have you looked at neighborhood order for the contact energies? See if that changes anything, try to give it a little bit more range.
Yeah, so in terms of the cell size, that wasn't something that we looked into. One of the things in terms of like Bruce's original model was it was, I guess, like a 100 by 100 with sort of like grid nodes effectively. So one cell would take up one grid node. So our thought to try and replicate that as closely as possible was to just have all the cells sort of like in Bruce's model be a set specific size. So again, in her case, 100 by 100 gives you what, 10,000 nodes. So the volume of the whole domain was 500 by 500. So the volume of each cell was set to be 25, so 1/10,000th of the whole range. So partly through trying to replicate her approach as closely as we could, we sort of left that unchanged and looked more at this lambda ball instead. But it would be interesting to look at it. Obviously, it would then move the CompuCell version of the model further away from the original model that we were trying to replicate. I mean, in terms of my PhD, the aim was to use my supervisor's model as sort of the within host scale of a multi-scale infectious disease model that would then be linked up to like a between scale epidemiological modeling framework as well. So sort of one of the main reasons for including this work was to try and provide some sort of cross-validation that says, yes, this framework is replicable. We can get similar results even with a slightly different framework. So therefore part of the aim with the CompuCell models to try and keep it as similar as possible. So yeah, as a result, cell size wasn't something that we really looked into all that much, but it would be interesting to see what impact it had. And then in terms of the contact energies, I think the neighborhood order I set for that was like order 5. I think one of the things that we wanted to was to try and reduce the sort of an anisotropy of it, so the tendency potentially of cells to just align with the axes directions, and sort of X and Y, and just everything winds up looking like sort of straight lines or looking like diamond shapes. So to give it a more realistic nature, we set sort of not just the contact energy, but like the neighborhoods for any of the processes quite large, based on the sort of literature to suggest, or some of the reference guides by the CompuCell developers, for what they've suggested in the past would be a large enough neighborhood size to try and reduce those effects. So we didn't actually explore that as a potential option, but that would also be an interesting thing to look at in future work potentially, or if anyone were to develop a sort of alternative version of our TV model in CompuCell going forward, it would be interesting.
And then could you postulate that there's a kind of coupling effect between the lambda chemo and the von Mises distribution? So really the contribution of the chemotactic attractant is just forming enough of a bias to drive the von Mises distribution towards the infection site, which is why the chemotaxis attraction is so invariant.
Possibly, yeah. I think our feeling in terms of why our model was quite robust to this was that ultimately, even with quite large values of the lambda chemo, the amount of energy that was then contributing to the Hamiltonian was ultimately quite small, especially if you were like an arresting macrophage that was quite a long way from the site of infection. The amount of chemokine that you would be sensing would be sort of multiple orders of magnitude smaller than a macrophage that's a lot nearer. So therefore, the energy contribution at that point would be almost negligible, even if we had a very, very large lambda chemo. So yeah, partly it will be an issue due to how much was secreted. I mean, that was obviously based on biologically realistic literature, but it does therefore mean that certainly you don't have to get too far away from the site of infection for that energy contribution to be quite small. So yeah, I think our feeling was that it made sense that the model was fairly insensitive to that, as even if we'd set it to huge values, the energy contribution to Hamiltonian would have been probably smaller than some of the other contributions. So yeah, there is a chance that the von Mises or the external potential contribution to the energy in the Hamiltonian was probably going to be bigger for any macrophages other than those that were right close to the site of infection, where the concentration hadn't sort of decayed to a low enough point where the macrophage was barely sensing it effectively.
I think we can move ahead to the next one, the next part.
Yeah, sure. Just to say, while we've been talking, I have finally seen phagocytosis occurred in his quick model run. So you can see now there's that sort of dark purple macrophage just above this big cluster of MTB. And we are now finally seeing some sort of release of chemokine concentration from that point. So, and yeah, in theory, some of the macrophages, once they get close enough, they will start to move towards it. But anyway, be interested to see if that finishes before 7:30. That's my time, 2:30 your time. Yeah, I'll quickly show you the code, and then I've got a few potential extension activities if anyone's interested. So cool. So just to quickly go through the code. See, these are all fairly basic. So it's a 5 by 500 domain. Obviously, it's 2 dimensions. So there's just one. The literature suggests that 2D representations of granulomas effectively capture the same results as 3D ones. It's just obviously a lot computationally quicker to do it that way. This just left us 10 by default. 30,000 steps corresponds to 200 hours, given that the step size is 24 seconds. That was based on like the quickest moving cell process, which is just a macrophage moving. These are all the cell types, as I went through in the talk. Some of the plugins that we've got, so obviously volume constraints. We did add in surface constraints, but we don't really consider them that closely. We do have neighbor track and pixel track, which are important, I suppose, for when the two cells are sort of in contact, or if there's free space for recruitment of macrophages from the blood vessels, and then especially boundary pixel track in that regard. So these are all the contact energies. As you'd imagine, with all these cell types, there are a lot. These are mainly chosen sort of phenomenologically to get the right behavior. So any with wall have this negative energy so that everything's repelled away from the wall cells quite strongly. Blood vessels were left quite high for most of these cell types to allow for there to be space enough that recruitment of new macrophages could occur. The macrophages have high energy with most cell types, but then have a fairly low one with the bacteria, so that phagocytosis can more easily occur. And yeah, and as I said, the neighbor order was quite high. This may be slowing down our code somewhat, but we wanted to have it high enough that cells weren't just moving biased sort of along the x or y-axis directions. The chemotaxis, as discussed in the talk, lambda was set to 2000. We have secretion of oxygen by the blood vessels based on all these different contact rates. They're reduced if they're within a granuloma. So granuloma in this case being defined as any of these 3 cell types. And then chemokine released by the infected and chronically infected macrophages. A diffusion, we had to call the solver multiple times because if the diffusion coefficient had been set to the actual biologically realistic value, it would have caused issues with the sort of the solver that was being used in terms of the numerical accuracy. So what we do instead for each Monte Carlo step is we call it a lot of times, and that therefore means that the diffusion is the right size, but the sort of accuracy of the solver itself isn't impacted upon. In our model, we have got oxygen decaying. That was the same as Roos model, so that's just matching in this case. And the diffusion reduced inside a granuloma, so any of these 3 cells. And then chemokine, similarly, we call the solver multiple times per Monte Carlo step to get the right diffusion coefficient. Then there is this decay over time, and then these boundary conditions just mean that it's we have this no flux boundary and just zero everywhere initially. Not much to see here. So the main thing of interest is the steppable, I suppose. So initially we build the wall to enforce the no flux. This sort of while loop sets up all the blood vessels so that they're randomly located within the domain. Pick sort of random coordinates and then build out a 5x5 blood vessel from there. And if you encounter a cell, then that's not going to be a suitable spot. Similarly with the bacteria as well, you can either have them set up centrally or randomly based on this parameter here, and then again search the space, and if there's a free enough space, then build up a 5x5 cell for the bacteria initially, and then keep going until you have enough bacteria in total. And then similarly with the resting macrophages, search the space and fill them in until you've got 105 resting macrophages to start with. We assign all the properties that we want to our cells, the target volume and lambdas. Our macrophages have ages so that they'll live the right length of time within these sort of minimum maximum lifespans, which is just randomly chosen. We also keep track of the angle of direction, and these lambda vec are going to be the things changed by external potential later on. The bacteria replication time are set within the lower and upper limits of the phenotypes, and then most of it is then captured in this growth steppable later on. So at the start, we just calculate the distance from the bacteria to the nearest blood vessel. That was mainly with checking to see if Ruth's conclusions from my paper held up, and then just save some initial conditions into these text files. And then as we go through the steps, look through all the different cell types. If it's a blood vessel, then we can consider recruiting a new resting macrophage every 150, so that'd be every hour. If there's enough space, then with some probability, which was set so that on average there's a 7% chance that you get a new macrophage every hour. Then if there's space enough, a new macrophage is added here. If we're looking at a fast-growing bacterium, then we keep track of the numbers over time, and they can replicate if there's space around them. So if there's enough space, then they can increase their volume enough such that they'll replicate after the correct amount of time steps. And then if we're far enough in and the oxygen concentration is too low, then they will change to a slow growing state, and they'll change their replication time accordingly, given that they're now in a dormant state. Similar with the slow growing ones, we keep track of how many there are. They can replicate as well if there's space enough around them, and if the oxygen concentration is too high, then they will change to a fast growing state and get a new level. The macrophages, if the resting macrophage encounters a bacterium, then it will become infected. And it keeps track of the number of bacteria it's phagocytosed in this ingested bacteria internal dictionary, and it will get rid of the bacterium it's just phagocytosed. And then every 10 Monte Carlo steps, it will get a new external potential force based on this von Mises distribution, and then lambda, vec, and y will be updated accordingly. It's similar for infected macrophages as well. We keep track of how many there are and how many intracellular bacteria they have. We set the lambda volume very high for most steps, but this is to ensure that the correct movements occurred. So obviously in CompuCell you can't set an actual direct speed for each cell type, so we just made the cells move at the right rate by just freezing them in place for most of the runtime effectively. And on the steps where we allow them to move, then they have the forces applied, again based on von Mises and the external potential. They will also do phagocytosis as well, and if they phagocytosed enough based on this internal dictionary, they become chronically infected, and if they've lived beyond their lifespan, then they will die, and they will release all of their intracellular bacteria wherever there happens to be space in this slow growing state, and then become caseum themselves. Similarly for chronically infected, again we enforce their movement rates by just setting lambda volume very high for most of the runtime. And then similarly they can phagocytose, and if we're above this burst size in terms of how many ingested bacteria there are, then or they've outlived their lifespan, then if there's space, they release all of their contents back into the domain as slow growing bacteria and caseum. These last bits are just saving some of the summary statistics effectively. The low frequency steppable is for oxygen consumption, as that's a very, very slow occurring process. So in order so that it happens regularly enough that something happens, and it's not so irregular that the sort of quantities involved are so small that sort of nothing's really happening computationally speaking. Every 10,000 steps, they uptake a certain amount of oxygen. We have the mitosis for the bacteria to divide once they've reached their doubling volume. And when they divide, then we have this update attributes to pass on to split the bacteria into two that are half the size of the doubling volume. So yeah, so that's the code. What I'm going to share with you now is going to be a toy version of the code, and then for the remaining time until 1:30, if you want, you can try and complete some of the activities that are suggested. Cool. So I'm now going to share the toy model should be available in the sort of student materials. I think under like today's modules, I've just uploaded this as a zip folder, so you can download it from there and then set it up locally on whatever version of CompuCell you've got running. It's essentially the same as the original, but I've simplified it slightly. So we've got rid of the oxygen, and we just have all the extracellular bacterium replicate between sort of the quickest fast-growing rate and the slowest slow-growing rate initially. I've also gotten rid of chronically infected macrophages as infected macrophages, and they're very similar. So initially we have this slightly simpler state. I've just noticed there's two in the chat. Sorry, Benjamin, possible essential immune modification. I see the use of vaccines, which this would be a time-dependent function. Sorry, Benjamin. Did I say that? I'm not sure. I've just seen your message in the chat. Yeah, in theory, we could incorporate vaccines that would potentially maybe change the immune response, or yeah. So in theory, we could perhaps consider vaccination from the start. I guess in some way we could incorporate it that potentially the immune response is slightly stronger potentially in some way. If I'm being completely honest, we haven't considered vaccination as an option up till this point. For most individuals, they tend to be vaccinated from a very young age. In most cases, like in the first year of life. I think it tends to be like a slightly sort of deadened version of the bacteria. So in theory, I suppose we could make it that the bacteria is there, but just possibly they'd all be in a dormant state potentially, or in some maybe sort of even slower replicating state, and it's easier for the immune response to take hold potentially. It's an interesting question. It's not one we've really thought about, to be honest. Yeah, the main thing that would be worth investigating, of course, is also adding in the adaptive immune response, so having T cells in the model, and also potentially incorporating antibiotics as well. So yeah, that would depend on the amount of time that's passed since the initial infection. But computationally speaking, it was taking too long to run simulations for long enough where realistically T cells showing up would make biological sense, or antibiotics showing up would make sense. So yeah, in terms of the activity, hopefully you can access the toy TB model that I think Marcus just posted a link in the chat. Obviously, it's quite a long running model, so it may not be possible to run it all the way through right now. So maybe just be worth trying to run it and looking at it for a few hundred steps and seeing that it works as expected. And then potentially as an activity, sort of the first thing you do would be maybe trying to incorporate chronically infected macrophages. So this notion that if they phagocytose enough bacteria, then they'll eventually burst. So, yeah, may want to add that into the code. Potentially as a further extension, you could add in, like, as one of the chemical fields, add in oxygen, and therefore then differentiate the extracellular bacteria. So for now, in the toy version of the code that you'll have, there's just one phenotype. So you need to change it so that there's now two, sort of a fast-growing state and a slow-growing state. And having some sort of upper and lower thresholds where if the oxygen is above this point, it changes to a fast growing, and if it's below a certain point, it changes to a slow growing point. So could add that in, perhaps experiment with different values for O high and O low, and see if that changes the amount of bacteria that change. you want to set the values such that not all the bacteria will change, but some bacteria could change. In Ruth's original model, they're sort of trying to experiment where some values would just lead to all the bacteria staying in the same state that they started, or if you set the values too high or too low, they'd all go to fast or they'd all go to slow. So trying to play around with that and find some middle ground where some change but not all do would be interesting, and seeing how sensitive the bacteria are to these thresholds. And then in theory, yeah, if there's time at the end and you're really interested, then you could add some of the extra things, like you could add in the adaptive immune response, so T cells and activated macrophages, and have some way that the T cells activate the macrophages. Potentially, in terms of recruitment, we just got it as a phenomenological thing, where for now it's just done on a probability. But potentially, if you're interested, you could add in tumor necrosis factor alpha. This is sort of in real life believed to influence the recruitment of macrophages to the site of infection. So could have recruitment, rather than just being some set probability, be dependent on the locally sensed concentration of this by the blood vessels. Much harder thing to do potentially would be to try and make the caseum liquefied. So that's one thing that's commonly observed in TB granulomas, is like caseum behaving like a liquid, this sort of dead necrotic material, and that therefore making it harder for like oxygen to diffuse through. So in our version of the code, we have it as like a frozen cell type, but trying to make it more of a liquid cell type. How easy would that be to do in CompuCell is something perhaps interesting that could be worth exploring. Although, of course, I appreciate we only have about a quarter of an hour. So, yeah, I guess in theory, try and maybe do as many of these things as you want. And if you have any questions, then I'm happy to try and help.
So outside, I mean, this is obviously what the paper's final conclusion was that the incidence rate for macrophages contacting this tuberculosis cluster sets the encapsulation probability effectively. Have you really gone through and tried to postulate outside of these random incidence rates, like proximity to the blood vessels or the macrophage contacts, really how to help drive these macrophages to find this initial clustering? Because running the simulation, you see that really if it doesn't get that random luck in contact, it really does get lost.
Yeah. In a sense, like how could like treatments try and enforce like macrophages to move closer, to mean, or more in terms of like a modeling sense, how to best incorporate that? Do you mean, sorry?
It kind of goes with both, right? Do you expect there to be some sort of search function that the macrophages could be implemented, and how to implement it in computational, or how do you drive it itself towards this infected regime?
Yeah, yeah, that's a good question. Yeah, I mean, I suppose, yeah. For now, obviously, in our model, yeah, we just have the macrophages move at random, and it's only when they sense this chemokine that they start to move towards the site of infection. I don't know if any research has been done on like how, or if there's a way of like enforcing macrophages to behave in a certain way, like, or if there's any like treatments or drugs that could be given on this front. Yeah, I mean, from a modeling point of view, I suppose also, like, I guess our model is quite a simplified version of what's going on. I mean, there are other cytokines involved. In terms of chemokines necessarily, I'm not entirely sure which other ones do influence it. But obviously there are the cytokines that have been found to sort of upregulate certain immune responses, downregulate them. So concentrations of those potentially may influence, maybe not necessarily like macrophage movement, but possibly like the activation of macrophages beyond just, in Ruth's original model, it was a simple case of like if a T cell contacts a macrophage, then it activates it. Whereas in reality, I think it's more a case of there's different types of T cells. Some of them have sort of a more cytotoxic effect. Will release like cytokines that help sort of kill off some of the macrophages, and others will help with the activation of macrophages. So there's slightly different types of T cells that aren't just doing everything in the sort of all-in-one sense that ours is. So possibly adding in more cytokines in future, maybe things I think like interleukin 10, interleukin 4. There's a few of those that have been implied to have an impact on how good or bad different immune responses are at any given point in time, sort of upregulating them when there's clearly a growing infection, and then sort of downregulating them again to sort of turn off their response. So adding in some of those might help with the macrophages. In terms of how well they sort of deal with the response. But I suppose again, that's their sort of aspects that possibly occur over longer time frames than what we've considered up to this point within the model. So yeah, sorry, I'm not sure if that answers your question fully or not, but yeah, no, it is something that will be interesting to add perhaps more biological realism going forward, certainly.
Yeah, the allegory for me is, I look at more influenza type infections sometimes, or wound healing, and you'll get things like alarmin or DAMPs basically driving them to the site due to the epithelial damage signal.
Okay. Yeah, I suppose for now all we've got is chemokine effectively sort of driving them there. But yeah, beyond that, certainly, yeah, there is I'm sure a lot more realistic behavior that we could incorporate going forward, which would it would be interesting to look into. A lot of similar work's been done by like Denise Kirschner's group at the University of Michigan. So I think Ruth's original model was in part based on that as well. And I think some of the versions of their model have a number of different cytokines that we haven't considered up to this point, but would be interesting to add in sort of going forward.
And if anyone has questions, feel free to unmute and ask or put it in the chat. We are at time, so I'll give a brief moment for people to ask any final questions or let you know if they're having issues with the code. I can attribute that the code did run fine in my local simulation.
Yeah, I'll just leave the QR code here again. If anyone did want to sort of read any further, then yeah, by all means, obviously feel free to email me later on if you have any questions beyond this session. Then yeah, more than happy to answer.
I will end the recording here in a second. I do want to thank you for presenting the work and then sharing the code. That is very cool work.